Hi. So welcome to this first part of the lecture on genetics of psychiatric disorders. So what I'm doing is I'm going to pre-record this and leave it available so in case the live lecture doesn't work. And what I plan to do is to go through a little bit about what we've come to understand about genetic variation and how it affects risk of psychiatric disorders, and also use that to illustrate a number of the methods of investigating genetic effects that have been applied at different times in different places. So hopefully that will, thinking of it from that point of view will be helpful as well. And just a brief introduction to say that there were very special reasons why we hoped that genetic approaches would be helpful for psychiatric disorders, because we had no idea what was causing them, what was going wrong with them. We didn't understand anything about the basic biology and physiology, and there was a hope that doing genetic investigations could lead us to the genes that are involved, then the proteins and the pathways and so on. And there was a special value for psychiatric disorders because the brain is very difficult to do other kinds of experiment on, unlike other physical tissues. So there was a sort of big idea that genetics held out a lot of promise for psychiatry. And one of the things I want to do today is to go through, some of how the methodologies have been applied, and just a brief recap of some of the methodologies that we use to investigate genetic contributions to disease in general. So first off, one can look at a family study, and we could see that cases of a particular disease tend to cluster in families, which suggests they might be genetic. We can also see that if there are increased rates of one disease, there might be increased rates of another disease as well. And that can suggest that there's a sort of genetic relationship or a similarity between the two different diseases. And those kind of approaches have been helpful in throwing light on the classification of psychiatric disorders. But moving on, those studies don't tell us anything about whether the disease is really genetic or not, because there can be other causes of familial clustering. And so people had to go on to do twin and adoption studies. A twin study, you compare identical and non-identical twins, and you see if the identical twins are more similar for the phenotype that you care about. And the idea is that the sets of twins share the same environments to the same extent, but identical twins, if they're more similar, it will be because they share all their genome, whereas non-identical twins only share half their genome. And the other method was the adoption study method, where you look at people who've been adopted at a very young age away from their biological parents, but if they still have the increased risk of the diseases that their biological parents had, then that's an indication that there's a genetic contribution to that that disease, because they haven't got it from their adoptive parents. So that gives you that there is a genetic contribution to disease, but it doesn't tell you anything about what the nature of that contribution is, whether it's one gene or many, Mendelian, dominant, recessive, what have you. And to do that, what you have to do is you have to look at the segregation, the pattern of inheritance of the disease through families, and that might tell you that it's an autosomal dominant disease or Mendelian, recessive, or whatever. But the thing to know about that is that either you see a clear mode of transmission or you don't. And if you don't see a clear mode of transmission, if there's a complex mode of inheritance, then it's very difficult indeed to say what's going on, whether there's one gene or many or several or lots of little genes or what have you. So for anything with complex mode of inheritance, you're in the dark until you can actually find localize some actual genes. So then in order to find those genes, you move on to do linkage and association studies. So linkage, you use markers to tag bits of the chromosome down through a pedigree. You find bits of the chromosome that are following the disease through the pedigree, and that gives you the notion that you have an approximate localization of the disease. And then what you go on to do is to do fine mapping studies based on linkage, disequilibrium, and association, which gives you a narrower localization and down to the level, hopefully, of an individual gene. Other methods that have been important in psychiatry have been to detect copy number variants, and I'll talk about that a bit later. And then what we've been doing more and more of the last few years is sequencing studies. So initially it was very difficult to sequence. We'd only sequence very small regions, genes, or even just little bits of genes. Nowadays, of course, we can sequence whole exomes, all the genes, or whole genomes, every bit of the chromosome with the genes, and the 99% that's not genes as well. And then that gets you down, hopefully, to the association between where you can see variants that are associated with particular phenotypes. But that's you've still got to sort of follow that through with kind of functional studies. You've got to, when you see a variant that seems to be always more often than not occurring with the disease, you then want to put that into a model system, an animal or a cell culture, and you say, well, what does that variant actually do? And how might it be, what are the pathways by which it produces the disease that we're seeing? So these are the kind of methodologies that we'll be using, and you'll see them kind of illustrated a bit. So I'll begin by talking about the two major mental illnesses, manic depression and schizophrenia. And I just really want to get across that these are really, really severe illnesses. These are completely outside the normal range of human experience. When you see people who've got these illnesses, when they're unwell, there's clearly something very wrong with them. And they're very important from a medical point of view. Each of them affects about 1% of the population. So these are, from a medical point of view, not rare diseases. We don't call these rare diseases, and they have tremendous costs for society, both in terms of the distress that they cause the patients and the people around them, but also actually in terms of other costs, in terms of social costs, economic costs to healthcare and to society in general, in terms of sort of lost employment and the mechanisms that are needed in society to support and help people with these conditions. So the first one was manic depression, or bipolar disorder, is clearly was always recognized to be a kind of strongly genetic condition with a big genetic contribution to its etiology. It was demonstrated formally by twin studies and by adoption studies, and you can see families in which there's a kind of quite a dense pattern of affection. So this was an Icelandic family that we used to carry out linkage studies, and what you can see is that the people shaded solid have got bipolar disorder, the people, which is episodes of mania and episodes of depression at different times. So people will be well for a while. You get an episode of mania when you're very kind of excited and overactive, maybe hallucinations, maybe delusions, and then episodes of depression is kind of like the opposite, lasting a few weeks or months, incredibly low mood, again, maybe delusions, hallucinations, scarcely moving, both very, very serious conditions. And the people who are shaded solidly in this pedigree had both bipolar disorder, and then there are also people in there that have just got unipolar disorder, so they only have the episodes of depression, and we think that that represents the same genetic susceptibility in these people. They just, for some reason, they're not developing the manic episodes as well. And you could look at that and think it's a bit like an autosomal dominant disorder with a major gene segregating through the pedigree. But the truth is actually that we haven't identified clearly that there are particular autosomal dominant forms of bipolar disorder. And there seems to be a complex mode of inheritance, which involves lots of genes, some with big effects, some with little effects. We tried to do linkage studies, but they didn't actually identify definite genes. And so when people moved on to genome-wide association studies, where you take a million common markers spread out across 23 chromosomes, then you do see places in the genome where you're reliably getting some genetic signal where one allele of a variant will be associated with bipolar disorder, and we use huge sample sizes, so we get very small p-values. So we're quite convinced that there are places on the genome where there is something going on which relates to the risk of bipolar disorder. The markers themselves are associated with very small risk effects, so it might be a cumulative effect of them, or it might be that they're tagging a more important risk effect in a rare variant nearby. And often it's hard to tell exactly which genes are affected, but there are some that sort of reliably come up. So not to remember the names at all, but just to say that CACNA1C is an ion channel. So it lets in calcium ions in and out of cells and is involved then in neuronal excitation. And these other genes, there's a few other genes as well that come up fairly reliably. So ion channel genes, synaptic genes, cumulatively, you can say these seem to be involved in bipolar disorder. You can take some of these, you can try to build a mouse model and say that if that gene's knocked out, then that has particular behavioral abnormalities. And people are sort of doing that sort of research, but I'd say there's nothing sort of really definite that's come out and say, oh, well, if you have that variant or that gene gets knocked out, then you get bipolar disorder. We haven't really got to that stage yet. At the moment, we're in the situation where we're carrying out big exome sequencing studies, and hopefully those will demonstrate some insights, but they're not, that we're not really any further than that at the moment. If we consider the related disorder, unipolar depression, so although you can get episodes of people with unipolar depression in these as relatives of people with bipolar disorder, in fact, unipolar depression is actually a much commoner disorder. much more multifactorial, much less heritable, some genetic contribution, but not much. People have been doing genome-wide association studies, and you do get sort of hits around the genome, but my own feeling is that there's not all that much in the way of biological insights that we're going to get from doing genetic studies of unipolar depression. I think what will happen is we will find results from studying bipolar disorder, then we'll somehow get a better understanding of what controls mood and how that can go wrong, and then we can take those insights into unipolar depression. But I think that I don't think the field of genetics unipolar depression is all that promising. So the other major mental illness is schizophrenia. Again, it has a similar prevalence, about 1%, very severe illness, causes a lot of disability, florid psychotic symptoms. Often people who have it need to carry on taking antipsychotic medication indefinitely in order to remain fairly well. And often they don't make a full recovery and find it difficult to work and sort of fulfill their potential. So it's a very kind of damaging disease. Now, unlike bipolar disorder, there are some suggestions of kind of important etiological contributions to schizophrenia, environmental risk factors, which are non-genetic, which are kind of around things that would affect normal development. And there's a theory in schizophrenia that it's a neurodevelopmental disorder, which means that although the symptoms appear in people in their 20s, actually something has gone wrong much earlier in the development of the nervous system. And that's feeding into that is the idea that if in utero, if a pregnant woman has viral infection, famine, malnutrition, stuff goes wrong during birth, then that can predispose to developing schizophrenia 20 years later. There's also reasonable evidence that cannabis smoking as a teenager or young adult can increase the risk of developing schizophrenia, although it's a bit difficult to sort out sort of chicken from egg there. So if people who are going to be developing schizophrenia smoke more cannabis, then it's kind of a bit hard to tease that about, but it probably is the case. Anyway, you do seem to have some environmental risk factors, but you also have a big genetic contribution to schizophrenia risk. And there was actually a real argument about that, and it still wages a little bit to this day with people who are not up to date with the literature. And it's really an argument about whether schizophrenia is a brain disease or whether it's a psychological adjustment to social stuff. And so whatever the reasons, there was a debate about it. People say it's not genetic. And actually, so people did very careful twin studies, adoption studies, showed that jolly well was genetic. And also there's a genetic contribution. I'm not saying determined genetically, but there's a major contribution to risk from genetic effects. And one of the ways this was done was by looking at the offspring of discordant monozygotic twins. So what you do there is you take a pair of identical twins, monozygotic twins, one of whom has schizophrenia, one of whom doesn't, and then you look at the risk of schizophrenia in their children. And the finding was that the risk of schizophrenia is very similar whether your own parent has schizophrenia themselves or is the identical twin of somebody with schizophrenia. And that's strongly suggestive that the risk from having a parent with schizophrenia is a genetic risk. It's not mediated by environmental factors, the fact they're schizophrenic. It's mediated by the genes that they pass on. So whatever, so people established that there was a major genetic contribution to schizophrenia. We tried to carry out linkage studies. It was all very exciting, it was very high profile, and because initial positive findings were not replicated, and then this kind of cast a kind of long shadow over psychiatric genetics and people saying that it sort of couldn't be done. And the first real concrete finding was a balanced translocation which co-segregated in a pedigree with schizophrenia and other mental disorders. So this is a Scottish pedigree, and everybody with a star under their symbol has got a balanced translocation between chromosome 1 and chromosome 11. The tips of the chromosomes are exchanged, and if you have two abnormal chromosomes, but if you get both the abnormal chromosomes, then you've got all the right genes, but not necessarily on the right chromosomes. And what you see is that everybody who inherited the translocation, not everyone got the disorder, but if you follow through the pedigree, you will see that far more than you could expect by chance, that people, very distant relatives who share the translocation also have a diagnosis of schizophrenia. And this was regarded as very convincing, still is regarded as very convincing, that in this pedigree, that translocation is increasing the risk of schizophrenia. And people looked at the gene which was broken through by this, disrupted by the, on chromosome 1, the breakpoint for the translocation, and they named that gene DISC1, disrupted in schizophrenia 1. And that was sort of the first gene for schizophrenia. However, that did not lead to a breakthrough in schizophrenia genetics. It wasn't the case that we then found out that lots of other people with schizophrenia had different variants in DISC1. It's proved very difficult to find out exactly what DISC1 does and how it has an effect on schizophrenia risk, and so on. But it was the first concrete finding, I would say, in schizophrenia genetics. Now, the next thing that happened was that people identified an association with copy number variants. So a copy number variant, to explain the terminology, is just a deletion or a duplication. So if we color code our 2 copies of chromosome 6, then usually we can see that every bit of chromosome 6, we've got 2 copies of it. But if you delete a bit, we've only got 1 copy. And if we duplicate a bit, we've got 3 copies. And so deletions and duplications are called copy number variants. And what was recognized was that there was a particular deletion on chromosome 22 responsible for a syndrome called DiGeorge syndrome or velocardiofacial syndrome. What became recognized was that a number of people with schizophrenia had this deletion that was otherwise extremely rare. And if you take patients with schizophrenia, if you look for this deletion, then nearly 1% of them will have this deletion, which to say is otherwise very rare. And if you look at it the other way around, if you take people with the chromosome 22 microdeletion and follow them forwards, then about 30% of them will develop schizophrenia. So it's a massively increased risk over the sort of background risk of 1%. And people followed up this finding, and they've identified about a dozen other regions on the genome where there are these very, very rare either duplications or deletions, but that if somebody has one, then it massively increases their risk of developing schizophrenia, so that you would say that that variant has caused the schizophrenia in that person. And again, taking it back the other way around, if you take a sample of 100 people with schizophrenia, then about 2% of them on average will have one of these copy number variants if you look for them. And there are people like me saying that this should now become a sort of routine diagnostic test for schizophrenia, because it will give you a sort of cause, and also some of these CNVs have other clinical implications. You might have risk of congenital heart abnormalities or what have you that would need to be sort of followed up. So the CNV did give us concrete causes for schizophrenia, but it didn't, again, unfortunately, give us massive biological insights, because typically one of these CNVs knocks out 20 or 30 genes, and it's proved very difficult for us to identify which of the genes that are important for giving the schizophrenia. And so here's an example of a few of those places, with the exception of one particular deletion, which on chromosome 2, which varies in size, and sometimes it's very small, and then it only affects one gene, and the gene that it affects is neurexin 1. So then we can see that deleting this bit of neurexin 1 looks like it's a risk factor for schizophrenia. And that kind of all makes sense in a way, because this is a synapse. This is where 2 neurons communicate with each other. So this is the presynaptic and postsynaptic neuron. So this will release neurotransmitter into the synaptic cleft, be picked up here, pass on the neuronal signal. And on the presynaptic membrane, a protein is expressed called a neurexin, which interacts with a postsynaptic protein called a neuroligin. And so then it's very easy to sort of think, oh, that's great. If you disrupt this neurexin with the CNV, then the synapse doesn't work properly, and that's what gives you schizophrenia, and schizophrenia is all about your synapses not working properly, which is kind of halfway true. But again, it didn't pan out as this massive, transformative thing, because it's not the case that lots of people with schizophrenia have got neurexin 1 deletions. It's not the case that if you look at all the people with problems in their neurexins, they tend to get schizophrenia. And basically, it probably is the case that this CNV knocking out neurexin 1 is a risk factor for schizophrenia, but it hasn't readily translated into a sort of broader insight into what goes wrong in schizophrenia. So the other thing that sort of people started doing in schizophrenia is genome-wide association studies, and there's been a number of these now. And essentially what they do, like genome-wide studies of everything, is they do develop hits. You do find places on the genome which are associated with increased risk of illness. But and you find lots of places, so it was over 100, it's gone up a lot now. But the effect sizes of each variant are very, very small. So you're talking about only an odds ratio that's very close to 1. So your risk of, it's 0.9 or 1.1, the odds ratio. So your risk of schizophrenia changes from 1% to 1.1%. It's a tiny effect, but it is telling you that there's something going on in the genome near that place. Often you can't tell which gene it is. You certainly can't say, oh, this variant has this effect and that's why you get schizophrenia. But it has, one of the things about GWAS is it said there's some variation affects schizophrenia risk. Another thing I'll show you in a moment is that was the schizophrenia GWAS, which led to the development of polygenic risk scores, which you now see all over the place in genetics. I mean, I don't regard that as a good thing, but that is something that's happened, and I'll show you those in a bit. And then the other thing that's also been happening lately is XNY sequence studies, which I'll show you a bit more of. So let's look at the GWAS first. So this was the 108 hit GWAS. So what you'll see is that this is the minus log 10 of the p-value here. So this hit here has got a p-value of less than 10 to the minus 30, and so on. There's a number of other, all these hits have got p-values of less than 10 to the minus 8 or 9, and these are sort of genome-wide significant hits. And this one that's the highest hit is actually at the HLA region. Now, as I said, sometimes it's kind of hard to say in general, you get a marker associated with the phenotype and you can't actually tell it's near some genes. It's not necessarily in a gene and you don't really know which gene is being picked up. But overall, statistically, the GWAS hits tend to be concentrated around genes involved in glutamate transmission. So the glutamate's a neurotransmitter, so that's kind of something going on there. And calcium channel genes, which we've already seen were implicated in bipolar disorder. And so something going on around glutamate transmission, calcium channel genes, and neurons and synapses in general. And that's the kind of thing that you get from GWASs. Now, another thing that you get from GWASs is polygenic risk score. So the idea of polygenic risk score is you take hundreds or thousands of variants which have very weak association with disease, but if you add them up all together, their cumulative effect, you get an overall idea of what an individual person's sort of PRS, polygenic risk score, genetic susceptibility to a particular phenotype is. And what you can do is you can then take that risk score, look at it and sample other people, and you can see that there's a very strong, not strong, but statistically highly significant association between the polygenic risk score and the risk of schizophrenia in a completely different sample. So these are the association, the p-value for that effect in different schizophrenia samples, and you see very, very low p-values. So there's definitely something going on with this cumulative effect of SNPs that you're picking up. It's somehow picking up something about the genetic risk of schizophrenia. And then you can do interesting things with it, like look at the polygenic risk score of schizophrenia and its association with bipolar disorder. And you see that there is still a significant association compared with other phenotypes like coronary artery disease, Crohn's disease, hypertension, and so on. So that's what that's telling you is that there's some shared genetic risk between bipolar disorder and schizophrenia. It's not telling you that they're the same disease and we should forget about psychiatric diagnosis, but it's telling you something about that nature. And people have taken these kind of insights and they've used other methods like LD score regression to look at the similarities in genetic risk between a whole range of different diseases. And so arguably that has been helpful in helping us change our thinking around some of these illnesses. And to give a sort of different example, as an aside, anorexia nervosa, the polygenic risk score for anorexia nervosa, or the equivalent, is associated not only with psychiatric diagnoses, but also with metabolic, like risk of obesity, so metabolic traits. So that gives you the idea that anorexia nervosa is not a purely psychiatric illness, but also involves a sort of metabolic susceptibility as well. The ability to get thin and stay thin is to some extent a metabolic ability. So you can get these kind of insights from doing genome-wide association studies. Now, I'll be the first to say that it's incredibly rare that you get any biological insight from genome-wide association studies. So I'm going to show you an exception where people have taken the genome-wide association study results and got some sort of run with it and got some sort of biological insight. So what it is if you remember, that says C4 variants, is that the biggest HLA hit was at this place called HLA, and there's a gene in that region called C4, which codes for component 4 of complement component 4, which is involved in the body's kind of inflammation and attacking foreign organisms and stuff. So it's the whole complement pathway, which is activated when you get an infection and a whole lot of other stuff. So C4 is near HLA. So what people did was they showed that there was variation in C4, and there was 3 different kinds of variation. First off, you could have allelic variation, a coding change would mention you either had C4a or C4b. Then you could have an insertion or not an insertion, and that made the gene long or short. And then you could also, on the same chromosome, have different copy numbers of the gene. So you can have 1 copy of the gene, or you can have 2 or 3 copies of the gene. So that could vary as well. So wherever you looked, each chromosome you looked at, you had a C4 haplotype, and you could see this kind of variation. And what was shown was that from the GWAS results, that there was a strong association between C4a expression and predicted C4a expression and schizophrenia, with the p-value is 10 to the minus 20. So I'll show you sort of how that was done. So first off, you can look in a small sample of a few hundred people, and you can sequence this region, and you can see which C4 haplotype they have. They can have a short BB, they can have a long, 2 long ALs, or 1 long AL, 1 long A, 1 long A and 1 long B, 1 long A, 1 short B. And what you can do is you can show that these haplotypes are associated with a different relative risk of schizophrenia, with an odds ratio of moderate odds ratio of 1.3. This is just shown in a few hundred people. You can also show in those people that these haplotypes are associated with different levels of expression of C4a. You get more C4a if you've got 2 long A alleles than if you have 1 short B allele. And you can see that the 2 mirror each other very well. You can also show, because this is a set of a few hundred cases and controls, if you actually measure the C4a expression directly, you can say that people with schizophrenia have a higher expression of C4a, but the p-value's rather modest. It's only 10 to the minus 4, so you're not going to get a paper in Nature out of that. And also, you don't really know with any of these expression studies whether the change in expression is causing the disease or is a result of the disease. So what these guys did was they took the GWAS variants and they used them to predict what the haplotype, each set of variants is associated with a different haplotype of the C4a. And so there'll be some haplotypes that will predict you've got 2 long A's, and there'll be a bunch of haplotypes that predict you've got a long A and a short B, and so on. So you can then go to your GWAS sample, which is thousands and thousands and thousands of people, and from that, from their SNP genotypes, you can say what you think their C4a haplotypes are and what level of expression of C4a they would then have. And then you can say, does that correlate with risk of schizophrenia in these thousands and thousands of people? And you can say, yes, it does. And the p-value, as I say, was 10 to the minus 20. So these were these guys going from GWAS SNPs, genotypes in thousand people, going to C4a expression in a few hundred people, and then taking the predicted expression back out into the GWAS subjects. And actually, from a biology point of view, it's not completely crazy that C4 should have something to do with schizophrenia risk, because it's present on dendrites and in synapses, that people with schizophrenia, there has been for many years an idea that something has gone wrong with synaptic pruning in schizophrenia. When in adolescence, there's a process called pruning where the number of synapses in your brain gets dramatically reduced. And one of the ideas is that that's gone wrong in schizophrenia. And there's suggestions that C4 could be involved in that pruning process. And also people made a mouse model and showed that if you knocked out the C4 in the mice, then they seem to have less synaptic pruning. So this is pretty, this is all new stuff. It's only just been happening, but this is the way, this is sort of one of the best bets of going from genetic studies into kind of plausible biology, and this will carry on being taken forward. So that was sort of from common variants. Now let's get back to sequence data. So people have been doing recently, the last few years, been doing sequencing studies. You can't see that gene, it says SETD1A behind me. And this was really the first gene where you could say if there's a sequence variant that knocks out the gene, not a CNV, but an actual sequence variant, then that is a big risk factor for schizophrenia. So it involves sequencing a few thousand subjects, including 1,000 trios. So what a trio is you have an affected case and you have both their parents. Now what that means is if there's a de novo variant, that's to say problem in meiosis, a new variant gets created, it's present in the child but not in either parent, then you can detect that the variant is de novo. And de novo variants are quite rare. So if you see a de novo variant, and if you see a de novo variant that knocks out a gene, and you see that person's got a particular phenotype, and you see other people with that knockout have also got the same phenotype, then you can be more and more confident that it's that knockout which has caused the phenotype. And indeed, that's what was done. So from a very small number of people who actually had loss of function mutations in SETD1A, so that's a stop mutation or frameshift mutation or splice type mutation, a very small number of people, only 10, we could still say very, very confidently that it was very unlikely that that would have occurred by chance. And people have accepted this result. And essentially, it does seem to be the case that if you have a loss of function variant in SETD1A, then you have a very high probability of developing schizophrenia. So 2 caveats about that. One is it accounts for almost no cases of schizophrenia. So it's incredibly rare. So it's not, oh, that's why people have schizophrenia. It's more the other way around. If you have the variant, you've got schizophrenia. Nevertheless, it could give you some useful biology because you say, oh, this is interesting. This biology going wrong gives you schizophrenia. But then when you look at what this gene does, it all gets a bit tricky because, you know, this was published, I guess, what, a couple of years ago, and your heart sort of leaps but then sinks because what SETD1A is, it codes for a histone methyltransferase, which I'll show you in a moment what that is. Now, in a way, we were reassured because there were already reports in related genes of other syndromes that involved intellectual development, not schizophrenia, but stuff that you could plausibly see that's something to do with brain development going wrong when you disrupt this kind of gene. But what does this kind of gene do? So DNA is wrapped around histones, and that's what makes a nucleosome. And the histones are these proteins that the DNA gets wrapped around, and the histones have tails which project out. And the idea is that you can methylate or acetylate or do things to particular amino acids in these tails, and that sends a sort of a signal which is read by the molecular machinery, which tells it whether to essentially to express the gene or to switch on genes, switch on more of this gene, less of that gene product, and so on and so forth. So it's called an epigenetic modification, and the idea is that methylation and acetylation can change gene transcription, which, you know, it's then you go, well, how on earth does that, changing that, messing around with that, give you schizophrenia? It's affecting all 20,000 genes in your body, isn't it? So it's a bit tricky, but presumably there's some kind of specificity. And this finding allowed people to build a mouse model. So this is just from last year, you know, showing what you do. You knock out one of the SETD1As in the mouse, and then you look to see what happens. And you can see expression change in a bunch of genes. You can see that the neuronal axonal development changes. You can see that synapses are different in the mouse brain. You can see that the mouse has deficits in its working memory. You can see some behavioral differences in the mouse. And also you can put in an antagonist of a methylase antagonist, I think it is, anyway, and try to reverse the effect. So this is very much work in progress, but the idea is once you find a gene, even though its function is obscure, even though it's incredibly rare cause of disease, then at least you can start building models, you can start impacting those models, you can start trying to understand what's gone wrong better and what you might do about it. Now, the latest in exome sequencing is only a preprint so far. We've got 24,000 cases, nearly 100,000 controls. And what that study is showing is 10 so far specific genes. So there are 10 genes that are statistically significantly implicating schizophrenia, SETD1A, and a few others. And below that, sort of not individually significant, implicating a whole bunch of other neuronal genes where what we are seeing is rare variants that are not picked up by genome-wide association studies, but that obviously can have big effects on functioning of individual genes, individually very rare variants, but when they do occur, having a big impact on risk of developing schizophrenia. And that's kind of the direction of travel. And from a psychiatrist's point of view, very excitingly, unlike the SETD1A, we're starting to see at least 2 genes which have a kind of obvious biological function. So they're genes which code for subunits of glutamate receptors. There's a whole lot implicating glutamate transmission of schizophrenia already coming from genetic studies. And now we're getting down to the level of individual genes, individual molecules. We can say, oh, that's interesting. If that receptor doesn't work properly, then that gives somehow increased risk of schizophrenia. Let's find out more about that. Let's do the mice models. Let's try out different drugs and see if we can affect its functioning, and so on and so on. So that's kind of where we are with the schizophrenia genetics. And then I just want to say, before I finish this first half, a little bit about alcoholism. So alcoholism, a few things really. So one is it's an example of a phenotype where there are genetic effects and non-genetic effects. So we're very aware of environmental risk factors for alcoholism, which include availability, price, occupation, so merchant seamen, doctors, culture. All these things affect risk of developing alcoholism. But also, if you do twin studies and adoption studies, there are clear genetic risks as well. So that's one thing about it. People are doing GWAS studies and so on, and there is sort of stuff popping up a little bit. But what I want to show you is one very clear little bit of biology, which is where we can relate a genetic variant to this complex phenotype of risk of developing alcoholism. And the genes that are involved are acetaldehyde dehydrogenases and hydrogenases. And the way it works is this. So there are variants in the some populations, East Asian populations especially, it's very common to have an acetaldehyde dehydrogenase mutation, which knocks out your acetaldehyde dehydrogenase. So this is how alcohol is usually metabolized. You turn wine into vinegar. So you oxidize the alcohol to acetaldehyde with alcohol dehydrogenase, and then you oxidize the acetaldehyde to vinegar, acetic acid, with acetaldehyde dehydrogenase. If you have a mutation in your acetaldehyde dehydrogenase, which in some populations is very common, then you can't do this step. And so you end up with circulating acetaldehyde. And you do not want to have circulating acetaldehyde. If you have acetaldehyde in your blood, you feel very unwell, you feel flushed, you feel uncomfortable. It's a very horrible sensation. And what that means is that you are less likely to drink alcohol. The alcohol becomes aversive, and you are protected from developing alcohol dependence syndrome because you drink less alcohol. And the same thing happens a little bit with alcohol dehydrogenase which is more widespread in the world. And there are some variants which make the alcohol dehydrogenase work more quickly. It's more effective. And again, people with these variants, then they make the acetaldehyde too quickly. And again, you can't metabolize it quickly enough. And so to a lesser extent, they also have flushing, and to a lesser extent, they also are protected against developing alcoholism. So you may think that's cheating a little bit to say that, you're protected against alcoholism by, you think I'm going to give you this gene for satisfaction or addiction or dependence or what have you, but actually it's just a gene for metabolizing alcohol. But I think it is illustrative of how, a biological variance can affect our risk of kind of complex behavioral phenotypes. Okay, so what I'm going to do there is I'm going to bring this part of the lecture to a close. So to sort of summarize progress in genetics and mental illness. So how things have changed with all this stuff over the last 30 years. I think we understand the illness is a bit different. We have sort of different views of it. We understand there's big variants and little variants. We understand that there are some shared contributions, some not shared between illnesses. We have, with schizophrenia at least, identified real concrete risk factor of big effects, copy number variants, and now finally we're starting to get towards individual genes, which will let us start getting to some biology, cell and animal models, new drug targets, and so on. So I'd like to think that with further sequencing studies, we'll make further progress. Some people might think I'm optimistic, but we're pressing ahead with it, and that's where we got to. So I hope you found that helpful. I'm going to break here, and then there will be a second part to this lecture, which I'll record separately.